conference last summer in Padua, in Italy, where was a conference on Xylem. And this was also a motivation that brought us to this work, which is to reconcile and to include soil physics into the current advancements that there are in plant hydraulics. And we started a few years ago together with Mathieu Javot to build this concept, and that's what I'm going to present you now. So the idea is to try to link. Let me see. I open the. So it's try to link below ground processes, so processes that they happen at the root soil interface, non-linearity of hydraulic conductivity around the roots, to the whole plants, and finally to the stomata and how they regulate the water fluxes. So, well, the motivation of this work is that it's fundamental to understand processes that control transpiration. And as transpiration dominates terrestrial water fluxes, we're talking about big water fluxes. So transpiration depends on atmospheric conditions and on stomata regulation on the short time. And stomata regulation indirectly depends on soil water status. So there is increasing evidence that soil moisture has an impact on terrestrial fluxes and on both water and carbon cycle. And understanding the mechanism by which soil drying impacts the stomata regulation and transpiration is an open, long-standing, and urgent question, I would say, in terrestrial ecosystem. And I think that soil physics can play an important role on this. Of course, this has implication on drought. Drought is a major constraint to crop production worldwide. And so this is relevant for agriculture. It's also relevant for forestry and ecosystem, as drought is a widespread cause of tree mortality across many forest biomes. And concerning trees, cavitation is a principal mechanism involved in tree mortality during drought. So far, concerning trees, a great emphasis has been placed on the relation between stomata closure and Xylem vulnerability. And there's been a lot of advances in this field, and the relation between stomata regulation and Xylem vulnerability is being used to categorize the water economy of plants, being them more conservative or more risky. So keeping stomata open or closing them to create a kind of safety margin before the Xylem cavitates. So the research question specific that we had is what are the mechanisms by which soil drying, and in particular here the soil water potential, what I call here psi soil, impacts the leaf water potential and the stomata conductance, and finally the transpiration. So the objective was to try to find a link between these four variables that I put it here. And when we do so, we have to acknowledge that actually there is not one soil water potential, there is always gradients, and the same is for the leaf. We have gradients also through the plants. So this psi soil has to be considered here as an equivalent soil water potential felt by the plant. And the leaf water potential, we can take it as the water potential in the xylem of the canopy, at some point in the canopy. So let's, I'm making it simple now, but we should acknowledge the gradients through these tissues. And the state of the art is that as the soil dries, the soil water potential decreases, and so decreases the leaf water potential. And the stomata regulation and the closure of stomata prevents an excessive drop in leaf water potential and then prevents that the xylem cavitates. And there are several papers, Brodrip, Schroeder, there are many works on this, showing this important relation between stomata regulation and the cavitation of the xylem. A fundamental work in linking the stomata behavior to the plant hydraulics and to leaf water potential, their dynamics, has been presented by Sperry in a series of papers. Here I'm citing one, but it's a long series of papers that Sperry and his co-worker proposed. But what is fundamental in this work is the idea that stomata close when the hydraulic conductivity of the xylem starts to decrease. And the idea is that stomata close when the relationship between the transpiration and the leaf water potential start to become non-linear due to xylem cavitation. And I think that this is fundamental because it makes a link between the opening and closing of stomata and all the plant hydraulics that there is below. So Sperry in this work put the focus on the cavitation. And his idea was that the main limitation to water fluxes through the soil, plant, and that to the xylem, to the stomata, and the main cause of non-linearity would be in the xylem. And the idea was that it, for him, was pragmatic to assume that there are enough roots to avoid big gradients in soil water potential around the roots. And then he assumed that then the xylem should be the limiting part, causing the nonlinearity of the system, and then the stomata regulation should correspond to the point of xylem cavitation. So plants should keep transpiring just before the point, or just before the xylem start to cavitate. So here I'm showing, it's a figure from Anderegg et al. 2017. It's a meta-analysis across species and biomes, and where it shows the relation between CX50. This is the xylem water potential at 50% of cavitation in the epsilon axis, and the CG is 50. This is the water potential in the leaves at 50% of stomata conductance. And when you plot these hydraulic traits, you observe that there is a correlation, there is a linear correlation, but all the points that are far below the one-to-one line. So the one-to-one line is the dotted line, and it's up here, and most of the points, they fall much below. And you can observe that there are points down there where the xylem is very resistant to embolism, but stomata, they close much before happen. So then the question is, okay, we had the hypothesis that stomata should respond to a decrease in xylem conductance, but how can they do it much before this actually drop in hydraulic conductance occur? And the hypothesis that we had in this work was that, well, we agree with the hypothesis that stomata close when the relation between leaf water potential and transpiration becomes nonlinear. But this nonlinearity is not caused always by the xylem vulnerability, but in many cases, this can be caused by the loss of soil hydraulic conductivity. And the hypothesis is that it is the loss of soil hydraulic conductivity more than the one of the xylem that drives the stomata closure during soil drying. Now, I want to make a short introduction to soil water flow, and I'll start with this figure by Xavier Dreyer from 2010, where they compare a series of soil hydraulic conductivities as a function of soil water potential getting more negative, where you have sandy soil, you have more loamy and clay soil, and they compare it to the typical hydraulic conductivity of plant roots. And what you observe in this picture is that, well, typically in wet soil, the soil is much more conductive than the roots, but when you start to dry the soil, the conductivity of soil that drops by several orders of magnitude, and eventually the soil is much less conductive than the root system. And this drop in hydraulic conductivity creates a gradient in water potential around the roots. These are simulations that Garner was doing already in the 60s, simulating gradients of water potential around single roots. And what he was finding is that in a wet soil, the gradients in water potential, they are almost negligible. But as the soil dries and the conductivity drops and the water potential decreases, you start to have big gradients of water potential around the roots. And they are caused by, well, the nonlinearity of hydraulic conductivity. So the closer you are to the root, the lower is the hydraulic conductivity. And due to the radial geometry of water flow to a root. When you approach a root, the velocity of water is increasing. And this causes big drops in water potential. And we can predict from simple soil physical consideration that there is a maximum transpiration that the soil at the given water potential can sustain. At that point, the soil can just not sustain anymore the transpiration demand of the plant. And the roots may start shrinking and lose contact to the soil. And this creates an additional resistance to root water uptake. And so nowadays, the resolution that we have in looking at the loss of contact between the roots and the soil is getting much better, and we can appreciate even better the big loss of contacts that you have around the roots. This is just an illustration of this picture, but we are doing it more quantitative. And in the past, I thought that the loss of contact to the roots was more the consequence of soil, so let's say of water limitation. But I'm starting considering it, and I think that the early formation of loss of gaps between the soil and the roots actually can be one of the causes of the nonlinearity that we will see and we will discuss later. So overall, what we discussed in the last slides is process that they happen down there, and these processes are strongly nonlinear, and there is a point where the soil cannot sustain anymore the transpiration demand of the plant. But if we want to understand the implications of below ground processes and rhizosphere processes to transpiration, we need to put this in the context of the whole plant. And now I simplify the system with four key hydraulic conductances or resistances, and all of these, they are variable. And one is at the rhizosphere, at the soil-root interface, and the soil. One is in the plant, and which depend on the anatomy of roots, aquaporin activity, and also anatomical development. Up is in the xylem, which may cavitate, so also there the conductance is limiting, and we are up there to the stomata. So ultimately, the stomata are the main resistance to water flow. Or let's put it in another way. They are, so it's the stomata conductance that determines the transpiration rate, together with atmospheric condition, leaf area, and so, but they are deciding how much water is transpiring. So the part below, the xylem, the root, the rhizosphere, they have to sustain the water demand. That depends on the stomata closure and on the atmospheric condition. But there is a link with what below. So the stomata conductance depends on the hydraulics. And as the soil dries, soil and root leaf water potential decreases, and this triggers the stomata closure through the active and passive signals that they involve ABA production in different sites of the plants. And it's mainly an active process, but the idea was this one that I wanted to make with this slide, is that what determines the transpiration are the stomata, but the stomata is controlled by the leaf water potential and the hydraulics, which in turn depends on what happens below. So we can put this key hydraulic conductance in a simple model. That this is the simplest model that one can think. and to describe the water flow from the soil up to the leaf xylem, up to the stomata. And we can describe the flow through the soil, through the roots, through the xylem with similar equations that are based on the relation between the flux, a gradient in water potential, and an hydraulic conductivity. And the key point is that all these hydraulic conductivities, they are variable. Now, for this case that I'm explaining and I'm describing today, I'm just considering the soil to have a conductivity that depends on the matrix potential, and the xylem to have an hydraulic conductivity that depends on the water potential. And they are illustrated here with a very, very simple parameterization, where I took the Brooks and Corey model for the soil hydraulic conductivity. Of course, you can use a more flexible model, Van Genuchten, dual permeability model, but this will not change dramatically the concept I'm explaining. And this would be the hydraulic conductivity of the plant that is constant and equal mainly to the root hydraulic conductivity, but not only, but let's say it's constant, until the point where the xylem cavitates and then the conductance of the plant is dropping. So you can put together this equation, and we chose the simple parameterization because then we can solve the problem analytically. And we thought that one way to plot the results of this very simple model is to show it in a three-dimensional graph, where on the z-axis we plot the transpiration rate, on the x-axis we plot the soil water potential in megapascal from saturation getting down to the wilting points, and on the y-axis the leaf water potential, also from 0 getting to more and more negative leaf water potential. Now, the model would predict that the plant would move on this surface. This two-dimensional surface is the hydraulic space where the plant will move. So let's say when transpiration is 0, so at pre-down, you will be on a one-to-one line. So the leaf and the soil water potential are equal. Let's say that we are in a wet soil and the plant starts transpiring. Then transpiration increases, and then the leaf water potential decreases, and this will follow a linear line. The slope of this line is the plant hydraulic conductance. Over drying, the plant will move on this surface, and this will be the diurnal rhythm. And let's say now we are in a relatively drier soil, we are approaching the wilting point, and when the plant starts transpiring, the slope is linear, but as the soil water potential decreases, the hydraulic of the soil decreases, and the surface starts bending. Then, because of the soil hydraulic conductivity decreases, so because the xylem cavitates. Any decrease of the hydraulic mass will make the surface bending. So you see that the surface is bent, wet soil at very, very pressure. So you may expect the leaf area will never, and the stomata conductance will be high to be there. You may expect the plant to be there. But when you reach a drier soil, the surface bends also at lower transpiration rates. So the hypothesis is that plants should not move into the nonlinear part of the surface for two reasons. One is because in that part, a slight increase in transpiration or a slight increase in stomata conductance would cause a nonlinear and very large drop in leaf water potential. Second is that you have to think that this is a temporal component. As soon as the leaf water potential starts to drop, then this will start to drop faster and faster and faster. So we marked in red the transition between the linear and the nonlinear zone, this line here. And the idea is that the hypothesis is that stomata regulation should prevent plants to cross this line. So we can look at the surface from 2 points of views. So we can look at it from the soil view, and this is what this is the transpiration rate versus soil water potential, and this is the typical view of a soil physicist, where we're looking at a decrease in transpiration over soil drying, and the red line is illustrated again in this figure. Blue line would be isolines in which let's say the soil water potential is constant, but this probably would mean nothing. So just see that the maximum transpiration rate that the plant could have is this one. there is nothing here because the soil would not allow it, or the hydraulic conductivity of the system would not allow it, and the red line would be an optimal behavior. These are typical lines that have been measured when you plot transpiration versus soil drain. The other view is the plant physiology view, where you plot transpiration versus leaf water potential. And the interesting thing of this line is that the red line, which is supposed to indicate an optimal stomata behavior according to the hydraulics, this would result in a transpiration in relation to the leaf water potential, where actually, to describe such a figure, you cannot have a stomata conductance that is simply a function of the leaf water potential. And it would also indicate that for the same leaf water potential, you may have at the same time a very high conductance and a very low one. So if we are consistent with this theory, we would find out that, well, the leaf water potential and the stomata conductance, so they should not be related by a simple function. And we will check if this is true later. Now, this was the theory which would suggest that we propose, and that was Perry was proposing, is that stomata maintains plants on the limit of hydraulic limitation, of hydraulic linearity. So let's come to some experimental evidences. These are experiments from the 80s by Pasura. He was doing experiments with the pressure chamber in which the soil and the root system were pressurized to maintain the water in the xylem of the leaves at atmospheric pressure. And in this way, the plants, the leaves stay turgid, and it can monitor over time the suction in the leaves. And what you can obtain with this type of experiment, you could build the three-dimensional surface. And this was an example again that illustrates that when you are in a wet soil, the relation between transpiration and leaf water potential is linear. And as the soil starts to dry, this relation becomes nonlinear. So actually, this nonlinearity, they can be measured and they can be well predicted also by a simple model. So what about the stomata? These again are experiments from the 80s that were done in Bayreuth, where I am now, and they were done in the group by Schulze. And in these experiments, he was at the same time measuring the plant hydraulics. These are the green lines and the green dots. So this was done again by pressurizing the plants. So seeing at what points the plant was turning, let's say, into the, so at what points the relation was becoming nonlinear. And this we indicated with the red line. And independently, he was measuring the leaf water potential and transpiration for plants undergoing soil drying for two vapor pressure deficit, high vapor pressure deficit and lower vapor pressure deficit. So what you see with the is that the red line is simply predicted based on the hydraulics, and it fits very well the stomata closure. There are not so many evidences of this behavior, and we couldn't find so many detailed measurements. So we took some, and the existing one, they seem to support this theory. But of course, we wanted to generalize it and extend it. And so we had a look at other works. And for instance, this is a recent paper by Brodrip and Rodriguez, Dominguez, and Brodrip on new phytologists. This is with olive trees. And again, they plotted the relation between stomata conductance, you see, this is the canopy conductance, as a function of the midday stem water potential. And again, what you see is this typical behavior of this relation, which reflects somehow what we expect based on the theory. So, and when I see such a curve, I would start thinking that, well, but then maybe it's true that the stomata is not simply function of a leaf water potential. Because you may have a water potential of -1 megapascal, where you have the stomata fully open and almost practically closed. So, and then we tried to go through a meta-analysis of data from Anderrek, where we plotted the transpiration versus delta psi, and delta psi is the difference between the leaf water potential and the soil water potential. And the soil water potential, in most of the cases, we took what was available, which is the pre-down leaf water potential, which might not always match, indeed. And according to the theory, if it's true that the stomata should maintain the plant in the linear zone, the relation between transpiration and leaf water potential should be a straight line. And this was for many plants, this was the case. Of course, you have a system where it doesn't work perfectly, a system where it actually fails. So we have to be honest with this. For those who measure these parameters in the field, they know also how difficult this is, and also the fact that plants are changing their conductance over time. They may complicate the situation, and looking at this figure is not as simple. But on the other hand, it's pretty striking that in many cases this seems to be confirmed. But let's take it as, again, as not a definitive evidence, and that's something that we still need to look into. But based on this preliminary observation, I would conclude that there are enough evidences or convincing evidences that stomata operate near the onset of hydraulic limitation. Now, the second question is, well, but what is this hydraulic limitation of the water flow from the soil to the leaves? Is it the xylem? Is it the root? Is it the soil? And I come back to the figure that I started from, the figure from Underreg, where they were plotting the water potential at which the xylem reached 50% of its conductivity, so the cavitation point, and the water potential at which the plant closed stomata to 50%. And the green points are the measurements, and they are much below the 1-to-1 line. There are cases where they're close, so there are cases where this matches, but there are many cases where you're much below. So we used the same model that I showed you before, in which we can fix a given xylem resistance, and we can put a different soil conductivity of different root length density, and this is so this is a plant with very long roots. This is a plant with shorter roots, and this will be a plant with even shorter roots. And we calculate it based on the concept before, the point of cavitation. And what you see is that actually the model can reproduce this data point very well. So it doesn't prove that the soil was the limit, but it just proves that if you consider the soil, you can explain these points very well. And then we try to say, okay, so let's try, because what is very, let's say, elegant with the theory of linking a point of xylem vulnerability with a point of stomata closure is that you can compare one value with another. And I thought we should try to find such a value also for the soil. And it's not enough to put, so a water potential where the soil reaches 50% of its saturation, because it means nothing. Because even at that point, the soil is much more conductive than the rest of the system. So we thought we need to find a water potential in the soil when actually this starts to limit the plant. So we thought to define a value called CS50 as the minimum soil water potential that can sustain up to 50% of the maximum transpiration. And we call it this CS50. And we thought, okay, then the real hydraulic limitation should be the maximum between the xylem or the soil limitation point. So we take the less negative one. And when we do so, all the measurements here, they end up on this figure here. And when the squares are solid, it's the soil that is limiting. And when it's open, actually it's the xylem that is limiting. So it can be both, but if you accept that the soil can be limited, you may reach a better coordination or a better fit between the point of stomata closure and an hydraulic limitation. So, and the stomata closure for most of plants, actually, they are closer to the point of soil hydraulic limitation rather than the xylem hydraulic limitation. So, again, this is not conclusive. I call it, it's a preliminary conclusion, but again, we have evidences, preliminary evidences or observation that suggest this one, that the soil more than the xylem that contains transpiration. Now, this may not be the truth for all the systems. And, but what I think it's important here is that if you accept that the soil can be the limitation, you open up a lot of important mechanisms that plants can have to control transpiration, to control stomata regulation, and to respond to soil drying. So, because plants can respond to soil drying not only by closing stomata, not only by closing this red line, but they can respond to this nonlinearity by changing the surface. And plants do it, and they do it in many ways. For instance, plants may avoid the nonlinearity by making the root system active, which seems counterintuitive, but it's plants do what roots. And the drying roots decrease their hydraulic conductivity through building anatomical barriers. Interestingly, it also with the diurnal rhythm. And this is by Caldeira and Tardieu that shows that plants that, I think he did this in maize that were exposed to soil drying, they were showing a diurnal rhythm of pouring with the aquaporin in the roots, down-regulating before the of transpiration. They would mimic or linearity to induce stomatal closure before the soil dries. So there the plants can control this surface and to reduce activity before the soil dries. But the other option is there are mechanisms that the plants can have to avoid the linearity in the soil. And the most evident one is to go more roots. But this is good to be done before the stress occurs. Otherwise, you need carbon, you need stomata, you need transpiration roots. So they need to be built in advance. Or plan work at the interface between root and the soil. And now I just slide to take some of these rhizosphere processes, but I will be pretty fast on this. But one relevant trait the roots have to decrease the water potential around the roots is the growth of root hairs. And root hairs, they grow into the soil pores, increase the contact of soils with the root surface, and they seem to do it in a very clever way. They grow randomly, but they seem to grow until they reach the contact and keep growing around the soil matrix. And this, let's say, they decrease distance that they want to flow at the root surface. So let's say root hairs to the end, there is an effective way to decrease the nonlinearity water potential around the roots. So it's a bit more complicated than this. So what we are learning, at least with, is that root hairs shrink, and they shrink before the whole root system seems to shrink, and before they have cavities. So it seems that actually root hairs, they are pretty vulnerable to tension in the water, and this is something that we are looking into. So actually, the mechanism of how hairs extract water from the soil and how they interact with the soil matrix, they are very complex. But we have evidences that they play a role at the plant scale in the relation between transpiration rate and leaf water potential. And these were experiments that I did with John Pasura using his methods, where we compare barley with and without hairs. These are random mutants, so we are same root architecture and root properties, same leaf area and stomata, maximum stomata conductance, and the only observable difference that we could report was the presence or not of hairs. And when we do not have hairs and plants reach a high transpiration rate, then they start failing very early. So at high transpiration rate, the leaf water potential starts dropping very early compared to plants with hairs. So the hairs, even if may shrink themselves, even they may lose turgidity and fail to sustain water, they still seem to affect at the scale in using an excessive drop potential. Another mechanism that may help plants to stay in contact with the root surface is something that I have investigated a lot last year, is the presence of mucilage in the rhizosphere. Here on the left, you have a picture creating. These are roots of maize before they reach the surface of the soil after a rain event, and when the atmosphere is saturated with vapor and the root surface is covered with a water film, you see that the gel around the root starts to swell and form these blobs around the root system. This material called mucilage is exuded at the root tip by most of plants, and it has several interesting properties that change the physical properties of the liquid phase in the rhizosphere. And particularly, make it more viscous. It makes the surface tension compared to water lower, and it also absorbs water. It decreases the water potential in the liquid phase. So altogether, the presence of mucilage has the effect to maintain the soil around the roots a little wetter compared with the bulk soil. So the soil dries, but the region around the roots stays a little wetter. And this was something that soil scientists and root scientists have observed since long, that layer around the roots seemed a bit wetter than the rest of the soil. Keep in mind that there is also strong hysteresis there. So the situation may be the opposite when after irrigation or after re-wetting of the soil. But another thing that we are learning that is very important is not only that the rhizosphere is wetter, but is the spatial configuration of water in the pores next to the roots. And these are some pictures. This is in the sand mixed with mucilage, and these are around the roots of a maize. And what you start to observe is the formation of filaments in the rhizosphere. And the question is, how can we explain these filaments there? And as an Italian, I had this picture in mind, and that well, when you have actually a viscous material, actually tends to make filaments, and compared to a liquid where the surface tension is dominating. Now think about two fingers next to each other and with some liquid in between. If you have water and you separate your fingers, actually the liquid breaks up at a critical distance. You have the same when the soil is drying and the soil pores stay constant, they don't move, but you have a critical point where the liquid phase gets disconnected. And this is due to the dominant role of capillary forces. And this is due to the Rayleigh instability. So, but if you have a viscous material, the filaments, it stays connected. And you can do the example with many viscous material. You can do with saliva. If you do it, you will see that you have long living filaments staying between the particles. And this is elegantly described by the Ohnesorge number, which is defined as the ratio between viscosity divided by the square root of surface tension, density of the liquid, and the characteristic length that for a filament you can take the diameter of the filament. When this number is big, the thinning of a filament or a capillary bridge between two solid particles, it changes over time, and it scales with the surface tension divided viscosity. This is the thinning rate of a filament. And keep in mind that mucilage is a polymer solution. So during thinning and during drying, the concentration increases, the viscosity increases, surface tension decreases, and then an exponential the filaments that eventually will never break. So overall, what I'm saying is that making the soil more viscous, the root is physically connected to the matrix in a similar way as they can do. And this is similar strategies to diffuse agents around the roots. So, summary of this presentation, where I proposed, it's not really a conclusion, we have evidences, but I am not conclusive, presentation that suggests the soil hydraulic activity more than the Xylem vulnerability trigger the closure of stomata, and it does it in a predictable way. This has important implication. When we accept that the soil may limit the transpiration rather than the xylem, so we open up, let's say, or we acknowledge many mechanisms that happen in the root soil system to attenuate these gradients. We have also other implications, and one of these is that stomata regulation is not simply a function of leaf water potential, but it depends also on soil and root hydraulic properties. And then probably this is more an outlook, identifying what mechanism actually allows plants to respond to a nonlinearity, I think is a fundamental question that we need to answer. This is from, let's say, from the point of view of a plant physiological understanding. And maybe from a soil physical point of view, we introduced this parameter like CS50 and to understand the point where the soil starts limiting transpiration. And I'm pretty convinced that we need a better definition of this. First of all, because we have gradients of water potential in soil, and so one soil water potential means nothing. And we will need to identify this point more clearly. Okay, that's all what I wanted to present today. Most of this work actually is it's inside this paper, which is open access, and it's more an opinion paper rather than a review and conclusive. And indeed, I believe that many things that I showed you today, they need to be proven, but I think it's an interesting framework to test hypotheses. That's how I put it. Okay, I think I'm finished with this one. I don't know, I may keep the slides on for the discussion.
Yeah, sure. Thank you very much, Andrea, for your talk. Anybody want to ask a question? Anybody?
Yes, I have a question. First, thank for the very interesting presentation, and the fact that the evidence that the soil, the water potential in soil is controlling transpiration is very attractive, and it makes sense in the mathematics. But you are talking not just about the transpiration, you're talking about the closure of the stomata. And my question is, how do the stomata sense the water potential in the soil directly?
Right. Oh, it's a fantastic question. Let's put it in this way. Okay. So I don't think that they sense directly the water potential in the soil. Well, yeah, so I mean, it's a long, let's say, I have to answer in a short way. So there are some evidences that there are, so let's say that, let's accept that most of this is mediated by ABA. And when I started this work, I was reading a lot of articles talking about the signal coming from the roots. This is mainly in the literature on crop physiology, but while discussing more and more with scientists working on stomata, I have been mainly told that actually the ABA is mainly produced in the leaves, in the mesophyll. And and this is a bit unlikely that plants feel the soil water potential in the soil. What is more likely is that by ABA, the plants feel the water potential in the leaves. But how could this then respond to a drop of hydraulic conductivity around the roots? Because actually our theory is more that the stomata respond to a decrease in conductivity somewhere in the system. And the fact that this is in the soil, probably for stomata makes no difference, could be everywhere. But now comes the next question is, how can plants feel a change, a decrease in conductivity? So I had a bit of theory that I may show it here. So, and the main thing is, how do plants respond to a change in actually a slope of transpiration versus leaf water potential? And I had this idea. And one idea is that there are two factors. One is the production of ABA, and this is proportional to the leaf water potential. But then we have a second factor, which is, well, initially I thought that this was the dilution of ABA by flux, by transpiration. And, but probably I was wrong on this. Maybe rather than this one well, let's say you have a plant that produce more ABA when the leaf water potential gets negative, but you also have mechanisms that they decrease the ABA concentration. And this could be the catabolism of ABA. It could be the loading of the ABA into the phloem, and that's why it ends up in the roots. It could also be the dilution of ABA, even if I'm not very, very convinced on this. Or it could be the fact that during photosynthesis, well, that ABA is not really function of the leaf water potential, but is function of the turgidity of the mesophylla. This is not really only related to the water potential in the leaves, but it depends also on the osmotic potential. Plants can adjust it. And I thought all these mechanisms may allow to feel the ratio between a leaf water potential and divided by either transpiration or assimilation. And if it was assimilation, probably this would be even smarter for the plants. And if you follow up this concept, you end up a figure like this one, where you have on the epsilon axis, let's say here is transpiration, but for a constant DPD, this could be stomata conductance. And on the x-axis, you have something like the concentration of ABA near the guard cell. And this is not only the production, it's something else. And what you would obtain is that below the red line, when the system is linear, you have that the slope is it's decreasing. This means that I open stomata and the ABA concentration decreases. But if I cross the line, opening stomata would induce an excessive production of ABA that is not, let's say, attenuated by loading into the flow or catabolism or dilution of fluxes. This is, it's not a theory that is very complete. And I think we have to, so for me, central in this theory is that the ABA production and the ABA concentration in the guard cell, if we accept that this is what is controlling the closure of stomata, this may not be simply a function of a leaf water potential, but there may be processes that may attenuate the buildup of FEA. And these processes, they may be accelerated when plants is doing photosynthesis and is transpiring linearly. Sorry for the long answer, but it's something I'm thinking a lot about it.
Anyone else? Just unmute yourself and ask.
I have a question.
Yeah, go ahead.
Yeah, I'm not hearing the question.
Yeah, I think someone's stopped asking. I'll ask the question. I have my mute. So I wanted to ask, one thing is, have you considered the plant hydraulic that is not related to xylem or the soil? You know, aquaporins and membrane permeability mineral is going to be regulated by the same agent that kills the stomata. So it makes sense to be regulated simultaneously.
Mm-hmm. Yeah, I mean, this could be easily entered in, where easily? It could be entered in the model and should be entered in the model. I mean, we didn't do it so far. So what do you think we should include?
So just like you took at least an empirical model of a xylem vulnerability from any paper. So there's empirical findings of, for example, leaf hydraulic conductivity, which declines before the xylem is blocked. And also the root hydraulic conductivity, part of it is definitely the disconnection from the roots, but also in hydroponics, if you add the abscisic acid, so the conductivity goes down.
Yes, yes, I agree. And I saw many recent literature that showing that before the xylem cavitates many other parts of component of the plant tissues, actually they are losing hydraulic functionality. I mean, you mentioned in the leaf. Also, there is a lot of shrinkage of tissues. And so in this recent paper that I was referring to from, I can find it again. So this paper by Brodrip, for instance, and Rodriguez and Dominguez, they were showing that the roots, the shrinkage of the root for them seemed to be a major resistance to water flows. I would agree with you that this should be increased. One example was this work by Tardieu showing that there is a diurnal rhythm of aquaporin regulation in the root system, which actually, very interestingly, for the leaf, they would feel like could be the soil drying or could be any part of the, and they would simply feel a decreasing of root. Then it's just to have so many parameters inside the model that then you lose a bit control on them. But I think it depends really on the question. Here the question was it's multiple. One may try to see, okay, is it true that stomata respond to some hydraulic limitation? And this is one study, and you may not check where is the limitation. You just want to see if this is something that happened. The other part is understanding what is the hydraulic limitation and find out, is it in the roots? Is it in some outer xylem part of the plant? Is it above ground? Is it below ground? And then one needs to really go in the detail. And yeah.
May I ask a question?
Yes. Hello.
Okay. Hi. If we take your model at its face value, then how come we have cavitation at all? Then if the stomata will close very early when this loss of hydraulic conductivity in the soil takes place, then how come we have cavitation at all? Well, they are not always protected.
Yes, yes, yes. That's actually, you're right. This sounds a bit inconsistent what I was saying, but in reality, stomata are not closing. They limit they reduce the fluxes. But when you are at very negative water potential, I mean, I can put these slides probably. For such plants, for instance, you may have cavitation. Well, it will occur indeed. So let's say this concept probably works to predict that the stomata start to respond to limits in hydraulic limitation a bit earlier. But then when the soil keeps drying, I mean, you still have some fluxes, and you, at low water potential, you don't need big fluxes to cavitate. So what this model would say is that probably if the soil is wet, plants would never go to transpiration rates that would cause cavitation. But as transpiration keeps, or let's put it in other ways, you not expect to have cavitation at when the stomata are 50% open or closed. You get it much later.
Thank you.
But yeah, yes. So thanks for clarifying it. Actually, it's a good point.
Okay, Andrea, thank you so much. It was great.
Oh, thank you all for listening, for the questions. Uri, do you want me to send the PowerPoint of the presentation, or you have it anyway recorded as you like?
I recorded it and I will put it on YouTube. I clearly share it with you and on Twitter. If someone asks me, I'll pass the question to you, and then I can share your trends paper.
Okay, super. Oh, I just wanted to mention again that this is a work that I did it together with Mathieu Javot, and we thought it over was for fun at the beginning, to trying to draw this three-dimensional figure, and we started 2 years ago, and well, I think what was very good was that it linked us to all the literature on plant hydraulics, and I think we benefited a lot from studies on that area. Okay, thank you very much.
Okay, thank you again, and bye everybody.
Bye everybody.
Thank you.
Thank
Bye.
you.